Okay, so now that we have talked a bit about how neurons communicate and the types of signals that are sent by different types of neurotransmitters, whether they're excitatory or inhibitory, let's sort of move on to talking about how all of this stuff sort of fits together in terms of understanding what's happening in the retina, and in particular talking a little bit more about within the visual system some of the differences between the rods and cones. We've already sort of talked a little bit about what some of the functional differences, well, a couple of the different differences, not just functional, structural differences between the rods and cones, in the sense that they just look different, right? In the sense that they're shaped differently, which is where their names come from. Obviously, they differ in terms of number. We have far more rods than we have cones. They differ in terms of their locations in the retina, right? Most of our cones are concentrated in that area of the retina called the fovea, whereas all of our rods are in the periphery. You have some cones in the periphery as well, but the vast majority of your rods are located off in the periphery. And those are some of the sort of differences in terms of structure and location of the rods and cones. But we also talked about some of the differences in terms of their functions as well. In particular, when we talked about dark adaptation, the fact that we primarily rely upon our rods when we're in the dark as opposed to when we're in the daylight. We talked about how rods and cones adapt to the dark at different rates as well, and which is what's responsible for that sort of two-stage process of dark adaptation. That very fast process at the beginning, which is the result of your cones adapting very quickly to the dark, but they sort of plateau very early on. And then that second stage, which is brought on by the rods kicking in and starting to adapt to the dark. They adapt much more slowly, but they continue on much longer than the cones do, and they end up being much more sensitive to light, which is what makes them more useful to being able to see in the dark compared to the cones. And so we're actually going to talk a little bit in this section about exactly why that is. Why is it the case that rods are more sensitive to light than cones are? And it has to do with some of the things that we've already talked about a little bit. But before we get into that, let's talk about sort of the structure of the retina again, and we've sort of said a lot of this already. Rods and cones are obviously the receptors in the retina responding to light, and they send signals vertically. So when they've been stimulated, they generate action potentials, and they send their signals through that neural communication process of generating an action potential and then transmitting neurotransmitters. They send those signals sort of vertically through the retina, through a network of cells, in particular bipolar cells, which we will come back to talking about, remember those, and ganglion cells and the axons of ganglion cells. And we've said that before, it's the axons of the ganglion cells that actually sort of combine together to form the optic nerve. But cells are not only sent vertically, right? So by vertical we mean sort of straight down from the rod or the cone. If this is a rod, right, talking about its signals being sent vertically down to, if this is a ganglion cell, that would sort of go out the back of the eye, forming part of the optic nerve that heads up to the brain. Signals are also sent horizontally as well, so between receptors. If there's another receptor over here, and then another one over here as well, signals can actually be sent not just in the vertical direction, but also in the horizontal direction. Between receptors, and we'll see that this is important. This actually is something that sort of gives contributes to some of the different functions between rods and cones, and it actually is something that ends up making rods more sensitive to light than cones, and also ends up explaining also why and something else that we mentioned before, that rods are more sensitive to light, but cones have better detail, better visual acuity, it's called. They're better at being able to see fine detail. We've sort of mentioned that before in the sense that we talked about how the fovea is the part of our retina where we have the best visual acuity, best ability to see fine detail. And we've said that the fovea is really the part of the retina where an image is focused when you're fixating directly on it. And so these sort of horizontal messages are sent via different types of cells within the retina, between bipolar and between ganglion cells by amacrine cells. So let's take a look at the retina. We've seen some pictures of the retina before. Up here, this top layer, are all of our rods and cones, right? And what did we say? That they send their signals, they send signals vertically through, so downward, through a network of relaying messages to things like this here. This one's actually these ones are all kind of highlighted. B here stands for bipolar cell. So it's sending its signals down through the bipolar cell, which forms synapses, not only onto the receptor itself. So this is a rod. This bipolar cell synapses onto that rod and then relays those messages to the ganglion cells here. So synapses onto this ganglion cell. And so here's an axon from that ganglion cell, which will combine with the axons from all of these other ganglion cells to form the optic nerve. So messages don't just go this way, they also go in the horizontal direction as well. And so here is a particular type of cell that is responsible for sort of doing that, transmitting messages horizontally between ganglion cells and between bipolar cells. So you can see this amacrine cell here extends its axons in sort of the horizontal direction. Okay, so it's responsible for sort of horizontal messages. Okay, so now that we've seen that this is sort of the layout of the retina, with rods and cones sending signals really sort of both vertically as well as horizontally. The reason that this is important is because it's contributing to something called convergence. There's a lot of convergence in the sense that signals from many rods are all sort of converging onto a few ganglion cells, and it's the same thing for cones. The signals from many cones are converging onto just a few ganglion cells, and we'll see why this is important coming up in a second. But what's interesting is that the fact that the retina is organized in the way that it is, with signals being sent both vertically as well as horizontally, contributes to some types of illusions. For example, this is something called a Hermann grid, and so this is a fairly strong illusion that occurs fairly easily in just about everybody. Probably what you notice is little gray blobs that seem to exist at the intersections of all of these white lines. But you notice probably that they disappear the moment you try to fixate on them. The moment you try to direct your eyes toward them, they're gone. So they're only in the periphery. So if you're looking here, fixating here, you can see three blobs in here, here, and here. Although the moment you try to look at them, they disappear. So this is completely an illusion. There's no gray blob that actually exists here. And yet, that's what we perceive, right? So what's different about this illusion from many of the other illusions that we've talked about and shown so far in class this semester is many types of illusions occur because of what we've called top-down processing, which is when perception is influenced and changed by prior knowledge, prior expectations, assumptions that your brain makes about what might be out in the world and those assumptions have gone wrong can lead to an illusion. Many illusions occur because of that, because of top-down processing, because of mistaken guesses, essentially, that your brain has made about what's out in the world, which can lead your brain to see, 3 dimensions when, leading your brain to see 3D when in fact you're looking at just a 2D image, is really an illusion of top-down processing. Whereas this is one of the sort of few illusions that occur because of bottom-up perception, right? Bottom-up processing, when perception is influenced by the stimulus that's out in the environment. And more specifically than that, this illusion occurs really because of the way in which the retina is organized, because it sends signals both in the vertical direction as well as in the horizontal direction. We'll talk more about that in a little while. So we've already said there's a lot of what's called convergence in the retina. And convergence is when lots and lots of rods and cones are sending their signals to just a few ganglion cells, and the ganglion cells are the ones that are receiving all of that vertical information, right? So you can sort of think about it this way, that if we have, a rod, if this is a rod, and this is a rod, and this is a rod, and let's say this is a cone, and here's another rod and a cone. I've drawn more rods than cones because we have more rods than cones. They send their signals vertically down to ganglion cells, right? Here's a ganglion cell. The axon of this cell will form part of the optic nerve. What do we mean by convergence? Essentially mean that although, as we saw before, there are other cells between the rods and cones and the ganglion cells, that essentially is what's happening is that the signals from all of these cells, right, all of these sensory neurons, the rods and cones, are sending all of their signals to just a few ganglion cells. So there's a lot of convergence onto this single ganglion cell. All of these messages from these sensory neurons are all converging onto this single ganglion cell. So there's a lot of convergence in the retina. About 126 million rods and cones converge onto just 1 million ganglion cells. And here we get to another difference between the rods and cones, in that there's actually a lot more convergence for rods than there is for cones. So on average, about 120 rods converge onto a single ganglion cell, whereas for cones, it's only about 6 cones converge onto a single ganglion cell. So there's greater convergence onto ganglion cells for rods than for cones. And there's absolutely, I guess you could say there's no convergence for cones in the fovea. There's actually a 1-to-1 relationship between a cone, a foveal cone, and a ganglion cell. What does that mean? That means that each cone in the fovea gets its own ganglion cell, and that ganglion cell doesn't receive any input from any other cones in the fovea. So it's a 1-to-1 relationship there. Okay, so I think the last thing that we'll have time for in this video is to sort of put some of this all together here in terms of talking about how all of this affects the activity of a particular cell. So, we've already said that neurons essentially communicate one of 2 different types of messages, either an excitatory message or an inhibitory message. And so the fact that there's lots of convergence, that's important because the activity of a cell, or whether or not a cell generates an action potential, depends on the total amount of signals coming from all other cells that are converging on that, right? So for any particular neuron, let's start up here, for example, this is showing how the activity of a single cell changes depending on the amount of excitatory input this cell receives, and as well depending on the amount of inhibitory input whether this cell generates an actual